I'm really excited to follow that talk because I'm going to be talking a bit about how we catalyze the next generation of protein function AI. So I'm Corey Hudson. I work at a nonprofit that's working on addressing the shortcomings in the field of predictive biology. We do this in two ways. One is through open data generation. We try and get a huge corpus of open data to provide to the community. And we also do public benchmarking. Public benchmarking is the big part I'm going to talk about today. So for those of you who know a bit about how generative models work, generative models have an internal statistical worldview representation of some set of phenomena. In predictive structural biology, that's a statistical grammar that uses evolutionary and structural physical constraints to say what's a feasible structure based on a starting seed sequence. So you take a amino acid sequence, you run it through this statistical representation, and out of the back end comes a feasible structure. Many times these are structures with no natural homologs. So this is a huge improvement. We went in the last few years from having about 120,000 structures that had actually been measured to now being able to do that with hundreds of millions of structures, and to the point where people actually don't do the structures anymore experimentally. It's trustworthy enough for a lot of classes. Oh, wow. So learning the structural grammar involved a couple things. My text has not shown up on this, so I will walk you through it. Most of this came from a process with the Protein Data Bank over a period from the mid-'70s till currently, really. And this was a process of generating a huge amount of data. This used to be handed over SneakerNet with floppy disks, and eventually was catalyzed in a competition called CASP. And the result of this was a huge outcome of a major protein model that won the Nobel Prize a couple of years ago. Oh, there it is. We had some advantages to this. One is it was a really well-defined problem. It was a match, eventually, between the problem structure and the ML capabilities. And we had a large, high-fidelity data set. And we brought a community together. That was actually maybe the biggest part, is a community coming here. But this is tens of billions of dollars of data, if you talk about how much this cost. The issue, that's really impressive work, and I don't want to in any way undercut that. But we want to generate proteins that are functional, not exclusively structurally feasible. This is how we create protein therapeutics. This is how we create enzymes that may be waste degraders or bioremediators, what we call valorizers, taking trash and turning it into valuable things. We need functional proteins for the next generation of bioengineering platforms. And it's how we go beyond using what's biologically available to what's biologically possible and really open up that field. But we've got a huge missing piece here. And the missing piece is that the models that currently exist for predicting function for proteins either use one of two approaches. They either do structural evaluation using a metric called PLDDT or TM score, which are basically measures of disorder, or they do a small scale experimental evaluation from a filtered set of generative sequences. These are the two approaches, and almost all studies out there use one of these two approaches. And the big problem we have is we don't have a comparative model for benchmarking and evaluation. There's no way of saying that if one group has created a model and another group has created a model that we even know who's doing better versus who has presented more captivating results. And so that's where we step in, and this is my little plug for my organization and for this type of science that we do. We bring in a huge amount of external collaborators around this problem. We hold workshops around this problem, and ultimately we have developed a set of technologies, techniques, and assays to work on this problem. And so I draw a lot of inspiration from ImageNet. I think ImageNet is one of the best things that ever happened to AI because it gave a standard for how you could measure image to text. There's a similar tool out there called Protein Gem, which works for deep mutational scans. The big challenge with that is it's over a huge amount of data sets, which are not exactly comparable. So it wouldn't be a biological talk if I didn't have an undefined acronym. And my undefined acronym in this is GROC-seq. GROC-seq is a technology that my group has worked on developing. It's a quantitative growth-based assay using sequencing, GROC-seq. And what it can do is generate per experiment about 500,000 protein variants at a time. It does that at the cost of about a nickel a variant. And so you can't exactly disentangle the economics of benchmarking from the actual measurement of benchmarking. And it happens at about a nickel, and we've gotten the process to about 2 weeks per experiment. And so we can do this over about 8 different protein functions right now. What this allows us to do is to critically evaluate protein functional generative models. We can take and then compare them on a single consistent platform that will say, generate 100,000 variants, generate 500,000 variants, generate 10,000 variants, whatever the budget is that we decide to set, and compare these across different models for generative function prediction. That way, we can do a couple things. We don't choose models based on their usability necessarily. We choose models based on their ability to perform well against the experimental task. Our hope with this is that this spurs innovation and it spurs competition. It allows people to come in with new, innovative, sometimes crazy ideas for how to redevelop models for generating protein function, and then compete against whoever they want and see where they fall. It allows more innovation in the community and allows us to build a community. In addition to the benchmarking, community building is another aspect that we see as pushing forward and catalyzing protein generation. In 2023, we had a protein engineering competition around amylases. We had 30 teams and about 1,200 unique sequences that were generated and measured. This year, we're redoing it for PETases, which is a class of proteins that break down plastic. We have many more teams than we had the last time. These numbers are not currently accurate. It's closer to 225 teams currently, and about 11,000 sequences, as well as 2 different tracks, one for prediction and one for generation. That's me. That's this work. Thank you.
Wonderful. Do we have questions and comments? Yes, in the back.
This is amazing work, by the way, sort of documenting and sort of building all of this data. It's really cool. How do you see it sort of transferring to sort of other data modalities within omics, whether it's metabolomics, single cell transcriptomics? There's already the human cell atlas that's already out there. But I'm just curious, how do you envision this model that you have built around benchmarking and building a lot of this open source data, sort of translating into other omics for eventually training of AI models? Thank you.
Yeah, I think it's a natural extension of this to take something and to be able to predict sequence, to then be able to have a structure and predict structural activity, or to say, for instance, instead of predicting growth, to be able to predict enzymatic features or protein binding features or transcriptional regulation features. I think the growth of this is enormous. I have a lot of hope for that.
Thank you so
Yeah,
much.
thank you.